Life on land is a uniformly desiccating proposition. Animals living on land always face a negative water balance owing to the large space occupied by evaporation in their water budgets. The organ-level physiology of the mammalian and avian kidneys offers an additional layer of adaptation that has made these animals exceptionally capable of adapting to a wide range of terrestrial conditions. This is because of the unique urine concentrating ability of the mammalian and avian kidneys, which has enabled the mammals and birds to radiate into all sorts of terrestrial habitats, ranging from mesic to quite xeric. There are several factors that go into the mammalian kidney's urine concentrating ability. Here's a coronal section of the kidney, as we've already seen, showing the cortex differentiated from the medulla. As you'll recall, this type of kidney contains three different types of nephrons. The cortical nephron, which has a very short loop of Henle, the medullary nephron, which has no loop of Henle to speak of, and the juxtamedullary nephron, which has long loops of Henle that extend from the cortex into the medulla. The ability to concentrate urine depends upon a complicated mix of interactions between these three different types of nephrons. We'll get into some of the details of that momentarily, but for now we can use certain gross physical measurements of the kidney as a rough indicator of the mix of these three different kinds of nephrons. In a kidney coronal section, we can identify a cortical coronal section area, which of course is proportional to the volume occupied by the kidney cortex. By the same token, we can measure the coronal section area of the renal medulla, which will also be proportional to the volume of the renal medulla. In both cases, these volumes will represent space that's occupied either by the medullary nephrons or, in the case of the cortical area, the number of cortical and juxtamedullary nephrons. The more space within the kidney there is to accommodate these different types of nephrons, the more nephrons there will be. There's one more measure we can make, and that is the thickness of the renal medulla section area. This thickness is an indicator of how long the loops of Henle of the juxtamedullary nephrons are. These measures of kidney structure are important because they're related in fundamental ways to kidney function. For example, the filtration rate is going to be proportional to the number of nephrons in the kidney. Each nephron contributes a certain rate of glomerular filtration to the kidney's total filtrate production. The more nephrons there are, the greater will be the kidney's total filtration rate and the total production of urine. The ability of the kidney to concentrate urine depends upon the loop of Henle. Because the loop of Henle is a countercurrent multiplier, this means that the longer the loop of Henle is, the greater will be the effect of the countercurrent multiplication. This translates into a greater ability to generate high osmotic concentrations within the environment of the kidney. Therefore, the longer the loop of Henle, the greater will be the kidney's ability to concentrate urine. Finally, because the reabsorption of water and the ability to concentrate urine depends upon the ability to do work, these functions will themselves be proportional to the metabolic rate. Remember that all of this is driven by active transport of sodium across the kidney tubules or the transport of chloride ions in the case of the loop of Henle. The more metabolic work the kidney can do, the greater will be its ability to reabsorb water and to raise urine concentration above the concentration of the blood from which it's derived. You can see the operational consequence of some of these things by plotting a simple measure of kidney structure, what we'll call the ratio of medullary to cortical area, against the so-called maximum U over P ratio, that is the ratio of solute concentrations in the urine over the solute concentration of the plasma. The larger this number is, the more concentrated the urine will be. A U over P ratio of 10, for example, means that the urine is 10 times more concentrated than the plasma. In the kidneys of fishes, amphibians, and reptiles, remember, this ratio can only rise to a maximum of 1. The urine can never be more concentrated than the plasma. With respect to the medullary over cortical area, the larger this ratio is, the relatively thinner the cortex is, as shown in these little cartoons here. There's a pretty tight relationship between the medullary cortical area ratio and the maximum U over P ratio. The proportionally greater this ratio is, the more the urine can be concentrated. The concentration ability is quite impressive. In kidneys with a very high medullary to cortical area ratio, such as in certain desert rodents like the kangaroo rat, the urine can be concentrated by as much as 25 times the concentration of the plasma from which it's derived. In humans, the maximum U over P ratio is on the order of 4 to 6. This relationship between the relative area of the medullary cross-section of the kidney and urine concentrating ability is a little odd, though, if you think about it. The kidney with a high medullary to cortical area ratio is a kidney that has loops of Henle that are relatively long compared to the size of the kidney. You would think, therefore, given the mechanism that we've just outlined for how the urine can be concentrated, that the concentrating ability of the kidney should depend upon the absolute length of the loop of Henle, rather than its length relative to kidney size. So, for example, you would expect that the kidneys of small mammals, which have small kidneys and therefore nominally short loops of Henle, should be able to concentrate urine to a lesser extent than should large mammals that have large kidneys and therefore nominally long loops of Henle. But in fact, the opposite is true. Concentrating ability of the kidney, as measured by the maximum urine over plasma ratio, actually declines with increasing body size. In fact, concentrating ability scales with body mass to an exponent just below minus 0.1. Large mammals, with their nominally long loops of Henle, cannot make more concentrated urine than small mammals, with their nominally short loops of Henle. This is just the opposite of what we expect from the simple model we outlined earlier, which predicts that long loops of Henle should enable the kidney to produce more concentrated urine. So, how do we resolve this paradox? To see the resolution, we have to be a bit more attentive to the physics of the problem. Here's our model loop of Henle, with filtrate coming in from the left, from the proximal convoluted tubule, and modified filtrate leaving from the right into the distal convoluted tubule. We're also putting in here the thick part of the ascending limb of the loop. We've been proposing so far that the loop of Henle helps to concentrate urine by setting up an osmotic potential gradient along the loop of Henle. The stronger this gradient, that is, the extent to which the filtrate's osmotic concentration can rise along the loop, the more concentrated will be the urine that's produced. It's actually a little more complicated than that because there are two things operating here. And to understand them, we have to understand the difference between a difference and a gradient. When we speak of the length of the loop of Henle, we're really using this as a proxy for the property that's really helping to concentrate urine, and that is the difference of osmotic potential that is set up along the loop of Henle. We designate such a difference with the Greek letter delta. So, for example, if we have a difference along the loop of 300 milliosmoles at the top and 2400 milliosmoles at the bottom, this is an osmotic potential difference that is 2100 milliosmoles. A gradient is something different because the gradient we are interested in refers specifically to a change of osmotic potential along the length of the loop. Whereas the osmotic potential difference has units of milliosmoles, the osmotic potential gradient has units of milliosmoles per unit length. If you had a 2100 milliosmoles difference between the bottom and top of the loop of Henle, and this was spread out along a loop that was 70 millimeters long, you would have an osmotic potential gradient of 30 milliosmoles per millimeter. We can now begin to see some of the thermodynamic issues involved here. What determines the osmotic potential gradient in the loop of Henle is the transport power that can be brought to bear on actively moving solutes across the tubule boundary of the ascending limb of the loop of Henle. We'll call this the transport power. The osmotic potential gradient will therefore be proportional to the work rate that can be brought to bear by the active transport proteins of the loop's ascending limb. The magnitude of the osmotic potential difference, on the other hand, is proportional to the loop's length, L. What does all this mean for the concentrating ability of the mammalian kidney? For one thing, it allows us to make some sense of the anomalous decline of concentrating ability with increasing body mass. Let's look first at the effective body size on the two parameters that we just outlined. If we plot the log of the length of the loop of Henle, we get a relationship that looks like this. Specifically, the length of the loop of Henle increases with body mass to the 0.13 power. That number will be significant momentarily, but for now, we can say that this will be proportional to the magnitude of the osmotic potential difference that can be generated along the loop of Henle. If we look at the power relationship, we have to decide first which metabolic rate we are willing to apply. For various reasons, it makes a lot of sense to apply the scaling of mass-specific metabolic power, which scales to body mass to the minus 0.25 power. This is sensible because the cells of small mammals have higher metabolic rates than do the cells of large mammals. This comes from many of the same factors that we spoke of earlier, namely that the tubule cells of small mammals are more packed with mitochondria, their mitochondria are smaller, and the mitochondria have more convoluted cristae. So if we're looking at the ability to power the generation of an osmotic potential gradient, this is going to decline with increasing body mass to the minus 0.25 power. This now makes the scaling relationship of maximum urine over plasma ratio to log body mass more sensible. As body sizes get larger and larger, the ability to power the generation of steep osmotic potential gradients declines. This decline in metabolic power exceeds the rate of increase of tubule length. So, for large mammals, the longer loops of Henle cannot be powered with sufficient energy to effectively concentrate the urine. In small mammals, on the other hand, even though their loops of Henle are nominally shorter, they can bring more power to bear on generating steeper osmotic potential gradients, which allows them to concentrate urine more. Mammals and birds are not the only terrestrial vertebrates around. Reptiles have also successfully colonized land. The kidneys of reptiles are not built to concentrate urine in the same way that the kidneys of mammals and birds are. Nevertheless, the imperatives of life on land are the same. There have to be stringent mechanisms for the conservation of water to offset the ongoing and substantial losses of water by evaporation to the air. The kidneys of mammals and birds conserve water by concentrating the urine. Reptiles conserve water by another clever means. Let's turn to that one now.